Let us explore a fundamental challenge in modern science. Do we currently lack the tools and instruments needed to fully probe, understand, and even program far-some equilibrium systems? These systems include energy-dissipating biological molecules, oscillating chemical reactions, feedback loops, and large-scale patterns of self-organization. Most of the physical world we study in the lab is close to equilibrium, stable, predictable, and relatively easy to measure. But life and many complex phenomena do not live in equilibrium. Cells maintain themselves by constantly using energy. Chemical reactions like the Belousov-Zhabotinsky oscillate instead of settling down. And ecosystems, neural networks, and even weather systems evolve through feedback loops. And these are called far from equilibrium systems, and they are much harder to study because they never sit still. You might ask, why can't we just use the same microscopes and detectors we already have? The problem is that our current scientific instruments are mostly designed to look at fixed structures or average behaviors. A traditional microscope might give a snapshot of a living cell, but it cannot easily track the dynamic, energy-driven processes that keep it alive at the molecular scale. Similarly, bulk chemical measurements average out the fluctuations and transient states that actually matter for understanding feedback loops and oscillations. Another challenge is control. Far from equilibrium systems are extremely sensitive. Small perturbations can lead to very different outcomes, a hallmark of self-organization. But we currently lack tools that can deliver precise, real-time inputs at the scales needed. Imagine trying to program a living cell to reassemble itself using existing technology. We can edit genes, but we cannot yet guide the dynamic molecular processes those genes control as they happen. There are promising directions, however. Advances in super-resolution microscopy now allow us to watch single molecules moving and interacting in real time. New approaches in quantum sensing and ultrafast spectroscopy are pushing temporal and spatial limits even further, letting us see energy dissipation events on femtosecond timescales. Microfluidic and nanofluidic devices can impose controlled environments where self-organization can be observed more precisely. But these are still early steps, and none of them can yet program far from equilibrium systems at will. Why does this matter? Because life itself is a far from equilibrium process. If we could fully probe and manipulate these systems, it could revolutionize biology, medicine, materials science, and even our understanding of the origin of life. We could design molecules and cells that assemble and adapt like living systems do. We might also predict and control larger-scale phenomena like ecological tipping points or the collective behavior of neural circuits. At this stage, we are still building the fundamental tools, microscopes for dynamics rather than snapshots. The frontier is not just about observing these systems, but learning how to steer them, guiding energy flow, reaction pathways, and feedback loops. Achieving that will likely require new combinations of physics, chemistry, biology, and engineering, and perhaps entirely new classes of instruments we have yet to imagine. Most physical systems we understand well are near equilibrium, where changes occur slowly and fluctuations are small. Far from equilibrium systems, by contrast, constantly exchange energy with their surroundings and exhibit dynamic behaviors like oscillations and self-organization. Examples include living cells, weather systems, and active materials. These systems are inherently nonlinear, making their behavior highly sensitive to small perturbations. Probing and programming them requires tools with unprecedented temporal, spatial, and energy resolution. Living cells continuously dissipate energy to maintain function, making them unpredictable compared to static crystals. It is like trying to study a flock of birds in flight compared to a single bird perched on a branch. Traditional scientific instruments, such as electron microscopes and X-ray diffraction, excel at imaging static structures, but struggle with fast, dynamic processes. Many far from equilibrium systems fluctuate at timescales ranging from femtoseconds to hours, spanning orders of magnitude. Capturing these dynamics without perturbing the system is difficult. Existing tools often provide snapshots rather than continuous monitoring. This limitation hampers our ability to observe causal relationships and feedback mechanisms. Electron microscopes can image individual molecules but often require freezing or fixing samples, missing live dynamics. It is like trying to understand a movie by examining only a few still frames. Biological molecules such as enzymes and motor proteins operate far from equilibrium, using energy to drive chemical and mechanical tasks. Their function depends on transient conformational changes that are hard to capture with current imaging methods. Single molecule tracking techniques provide some insights but are limited in throughput and perturbation-free observation. Understanding these processes at the quantum or atomic level requires breakthroughs in detection sensitivity. Without this, we cannot fully model how energy dissipation controls life. ATP synthase spins to produce energy in cells, but its nanoscale rotary mechanism is difficult to track in real time. It is like trying to watch the gears of a running watch without opening it. Chemical and biological systems often exhibit oscillations driven by feedback loops, such as circadian rhythms or predator-prey cycles. These systems can switch between multiple states, amplifying small disturbances into large effects. Instruments that perturb the system can disrupt the natural dynamics we seek to measure. Capturing how feedback loops shape function requires minimally invasive sensors and precise perturbation tools. Current methods often disturb the very phenomena they aim to study. The Belousov-Zhabotinsky reaction shows oscillating colors as chemical concentrations shift cyclically, but probing it can disrupt its pattern. It is like trying to measure a soap bubble's thickness with a needle. The act of measuring pops the bubble. Self-organizing systems form complex patterns from simple local interactions, seen in tissues, colonies, or nanomaterials. These emergent behaviors are challenging to predict because they involve many interacting components across scales. Existing instruments often measure either fine details or large-scale patterns, but not both simultaneously. Without multi-scale tools, it is difficult to connect molecular interactions with emergent phenomena. This gap prevents us from programming self-organizing systems with precision. Actin filaments in cells self-organize into dynamic networks, but tracking their reorganization in 3D is extremely hard. It is like trying to watch every player and the whole strategy of a football game at once. We need tools that can capture energy flows, molecular conformations, and interactions in real time without perturbing the system. Emerging techniques such as quantum-enhanced microscopy, ultrafast spectroscopy, and integrated sensors show promise. These must be combined with AI-based analysis to interpret complex datasets. Developing programmable probes that respond adaptively could revolutionize the study of dynamic systems. Without such innovations, our understanding remains fundamentally incomplete. Quantum sensing using nitrogen vacancy centers in diamond can detect magnetic fields at the nanoscale without invasive contact. It is like using sonar instead of diving underwater to map a reef without disturbing it. To move beyond observation, we must learn to control and program these systems. This requires tools that can apply energy, chemical signals, or fields with fine spatiotemporal precision. Synthetic biology and active materials research already aim to write new functions into living and non-living systems, but the complexity of feedback and noise makes precise programming difficult. New instruments must integrate sensing and actuation in closed loops. Optogenetics uses light to control cellular processes, but tuning dynamic behaviors remains difficult. It is like conducting an orchestra without being able to hear how each instrument responds. Cracking far from equilibrium systems could transform medicine, energy, and materials science. We could design cells that self-repair, catalysis systems that mimic life's efficiency, or materials that adapt autonomously. Understanding energy dissipation and feedback loops would also advance climate science and ecosystem management. The tools developed would likely overlap with quantum sensing and machine learning, impacting many disciplines. This challenge is both scientific and technological. Being able to control protein folding could lead to cures for diseases like Alzheimer's. It is like learning to reprogram a computer at the hardware level instead of just changing its software. We are at the edge of a new scientific revolution, awaiting tools that can probe the most elusive systems in nature. These systems hold answers to how life sustains itself and how complexity arises. Bridging the gap requires interdisciplinary collaboration across physics, chemistry, biology, and engineering. The instruments we lack today could be as transformative as the invention of the microscope. Until then, far from equilibrium systems remain among the most mysterious phenomena in science. Just as the electron microscope revealed viruses for the first time, future tools may reveal hidden rules of self-organization. It is like exploring a continent hidden behind a fog bank. Once the fog lifts, a whole new world will appear.